Hi, Biology 158. Welcome to the mini of sorts unit on microbiology. So there is no official chapter dedicated to microbiology within your text. So for this section of the course, just be focusing on this PowerPoint and this lecture that I'm giving in terms of everything you need to know for exam number 2. So with that, we'll go ahead and get started. So for microbiology, we're basically just talking about all the different kinds of microbes that exist and giving a brief overview of each of them. So we define a microbe just as a microorganism, micro meaning small, so it's just a very tiny organism, something that may or may not be seen with the naked eye, but most likely not. This encompasses a whole bunch of different things. So we have your bacteria, we have a bunch of different kinds of protists, we have viruses, as well as the archaea domain. So for all the microbes within the tree of life, we have 3 big domains. We have domain bacteria, which house your bacteria. We have domain archaea, which house your archaea. And then we have domain eukarya. So eukarya by itself is not the domain that has microbes in it. Specifically, we have the kingdom protist, which have some microbes in it when we get further in with the taxonomy. Then on the sort of periphery, since it's technically not considered living, we have viruses. And we are going to talk about each one in turn. So microbes are very, very successful because of their genetic diversity. So we have things like bacteria and things like archaea that have a lot of similarities in their structural design and reproduction. However, they are able to live in completely different places because in part their differences in genetics. So they are very, very genetically different despite looking similar. Same idea with viruses. So viruses can live in lots of different environments. Despite being and having some similar structures, they also are very genetically diverse. So you can have things like your E. coli live in your gut and help with digestion. You can also have types of archaea that live in really extreme environments. So things like your geothermal pools that are ridiculously hot, or really salty oceans that not a lot of other stuff lives there. So a lot of different places that microbes can live in to be successful and inhabit areas where there aren't other living things. So first things first, we're going to start with bacteria. So we really just define bacteria as prokaryotic cells. So this means cells that don't have a nucleus or membrane-bound organelles. And just for a quick recap, membrane-bound organelles mean structures like your endoplasmic reticulum, your Golgi complex, and your vesicles. They're all part of packaging things up and shipping them out of the cell. So none of those structures are associated with prokaryotic organisms, of which bacteria is one. So bacteria are single-celled prokaryotic organisms, and they have a very simple internal structure. So mostly, you always have the bacteria with some kind of cell wall and capsule, so to basically contain the cell and give it structure. You also have a plasma membrane for allowing substances to come into and out of the cell. Then you'll have some kind of DNA with the chromosome in a ring, and then you're going to have ribosomes, so for protein synthesis, since all organisms need to be able to make proteins in some capacity. And then something kind of special for bacteria is that you're going to have what are called plasmids. These are separate rings of DNA that are separate from the chromosomes, but they serve a lot of important functions. Those plasmid sequences give rise to specific proteins to carry out certain functions for the bacteria. And then to move throughout their environment, they're going to have a flagellum or a whip-like tail. So they have all these structures, but they don't actually have any form organelles. Again, things like your endoplasmic reticulum, your Golgi complex, vesicles, vacuoles, all those things you learned about in intro bio. And we commonly define bacteria by their shape. There's 3 distinct shapes we have for bacteria. We have cocci, which is just spherical. We have bacilli, so these are just your rod-shaped bacteria. And then we have spirilla, which is like a spiral. Bacteria are kind of weird in that they can reproduce in a manner that's somewhat sexual, but also asexually. And we'll talk about both the sexual reproduction and the asexual reproduction. And then we can also classify bacteria based on how they metabolize energy. So for bacteria, we have what are called chemoorganotrophs, and these are just bacteria that can basically get their energy from organic molecules, much like you and I. So something like carbohydrates is something that they break down for energy. We do something similar. Bacteria can also utilize inorganic molecules, and those are called chemolithotropes. So they can utilize things like iron or hydrogen gas or ammonia to make their energy, which is actually really cool because that's not something a lot of organisms are able to do. Many organisms have to have some sort of organic molecule to make their energy, but some bacteria are very capable of using non-living materials or non-organic materials to get energy. Then we have some organisms or some bacteria that are called photoautotrophs, and these are able to utilize photosynthesis, like many plants, to produce their own energy to then break down for ATP. So an example of that is your cyanobacteria. These live in a lot of your aquatic ecosystems and are able to utilize photosynthesis. And then lastly, we can sort of classify bacteria based on if they are aerobic or anaerobic. So remember, aerobic means that they need oxygen as part of their metabolism. And then anaerobic means that they don't need oxygen for their metabolism. It's not an easy breakdown. It's some chemoorganotrophs can be aerobic, some can be anaerobic. You can have some chemolithotropes that are aerobic and some are anaerobic. So the line is kind of blurred in that way, but it just really speaks to the diversity that we have of bacteria in our world. So with bacteria production, there's sort of, again, two ways that this can happen. It can happen through asexual reproduction and a form of sexual reproduction. But we know that bacteria carry their genetic information into the two structures I pointed out a second ago. So you have that large chromosome, usually a singular one, that has the majority of the genetic information for a bacteria. Then you also have what are called plasmids, which are smaller circular DNA molecules that have, again, certain genes for specific functions for the bacteria. And the way that bacteria reproduce asexually is called binary fission. So this is pretty much akin to mitosis for our cells. We're producing a genetically identical copy. So with bacteria, we'll have the bacterial cell with the chromosomal DNA and then the plasmid DNA. We'll have that DNA get replicated, same way it happens in mitosis, where we have the S1 phase, or sorry, the S phase, where the DNA gets synthesized and copies itself. So we'll get a copy of the chromosomal DNA, and then we'll get copies of the plasmids in the bacterium. Then we'll have cytokinesis, so that cytoplasm will divide, and then one chromosome and some of the plasmids will go to one side, and then one chromosome and some of the plasmids will go to the other side, then having two daughter cells, which are genetically identical. That is binary fission because those offspring have the same genetic information as the parent cell. Then we have what's called lateral gene transfer, and this is the sort of sexual reproduction that I put in air quotes, or I put in quotes on the previous slide. So when we talk about sexual reproduction, we talk about a mixing of genetic material. So for animals, for example, we have sperm from the father that carries dad's genetic information, and we have eggs from the mom that carries her genetic information. We then have the sperm fused with the egg. There's mixing of genetic material, and that then grows into an embryo and a fetus, then giving life to a new being. Bacteria don't have sexual reproduction in the traditional sense. What they do is they do have a mixing, however, of their genetic material, which still sort of classifies it as sexual reproduction. And again, we call this lateral gene transfer. They're not actually making a new bacteria, but they are mixing genetic material and creating genetic diversity out of this mixing. And this can happen in three different ways. The first one's called conjugation. Conjugation happens basically when one bacteria creates a bridge linking to the other bacteria, and then basically passes some of its DNA, the plasmid DNA, from the donor to the recipient. So then that recipient can then incorporate some of that plasmid DNA into its plasmids, giving it new genes. We then have what's called transduction. And transduction, instead of a bacteria donating the genetic material, we're going to have a virus basically inject its DNA and have that DNA get incorporated into the plasmid. And then lastly, we have what's called transformation. So transformation is when we have the DNA fragments from some sort of broken down bacteria or other cell in the environment basically get engulfed by the recipient bacteria, and they can then incorporate that DNA fragments into their plasmid. So in all three cases, we are having new genetic material get introduced into the recipient bacteria, which is mixing with their original DNA, creating new combinations and creating new diversity, thus making it sexual in a sense. So bacteria have the ability to be both helpful for us and to be harmful for us. So on the helpful side, we can have bacteria in the body that help prevent infection from harmful bacteria. We have what's called probiotic therapy, and this is basically when we introduce a bunch of benign or non-harmful bacteria in really large numbers to sort of swamp or get rid of the harmful form. So we're basically overcrowding it and getting rid of the bad ones. Additionally, bacteria is in in some of our foods, things like yogurt that we eat regularly help with good gut health. But on the other hand, bacteria or a subset of bacteria can be harmful through what are called pathogenic bacteria. Basically just means they carry disease. And this can run the gamut of being mildly inconveniencing to being a life debilitating condition. So something like food poisoning, which can be caused by certain kinds of E. coli from eating spoiled food, can ruin your night or a couple days, but you'll recover from it. But then we have bacteria like chlamydia and gonorrhea from sexually transmitted diseases, which cause a lot more problems on individuals as well as healthcare. So now we're on to the second group, which is called archaea. So we're not going to talk too much about these because these are kind of a little more abstract, and we don't talk too much about them in terms of their relevance to A&P, but it's important to know about them. So archaea are very genetically different from bacteria, despite having a lot of these similar structures as bacteria. So they have plasma membranes and cell walls and flagella and all those structures I talk about with the bacteria. Also, like bacteria, they don't have a nucleus or nuclear membrane. However, despite looking and having many similar features to, they are actually very genetically different from bacteria. Part of this genetic diversity allows them to be living in very extreme environments. We often refer to archaea as what are called extremophiles. They can live in very, again, for lack of a better word, extreme conditions that make them really hard to get to and hard to study. For example, something like a volcano could have archaeobacteria or archaea. Again, a very salty ocean or a very deep ocean with a thermal vent, thousands of leagues below the sea, could house archaea as well. So they're kind of tucked away in really hard to reach areas for individuals to study them. However, despite this, they are living microbes and they are part of our ecosystem. Then we have our protist. So for our protist, this is within our domain eukarya, and our protists don't neatly fall into a sort of good taxonomic group. We define protists as kind of the everything else group that doesn't fit into something like an animal or a plant or a fungus, so on and so forth. But we can sort of define protists by how similar they are to certain groups. So we have animal-like protists, we have fungus-like protists, and we have plant-like protists. Protists are not only microbes, they can also be macrobes, so they can be large visible organisms and they can be small organisms as well. We're going to focus on just the microbe side. So first ones are called animal-like protists. And these animal-like protists are mobile, so they're able to move around their environment. They're not stuck in one place, and they actually hunt for their prey, much like animals do. A big group of these are called urciliates, and urciliates are named so because they have all these little hairs on the outside, where I'm running my mouse, called cilia. And in their many times aquatic environments, they can propel themselves through the water using their cilia, like a bunch of little oars. And because they hunt for other organisms, they are going to feed on them by a process we have covered many, many times in the immune chapter, phagocytosis. Hopefully by now you're very familiar with that term, where we are engulfing a smaller cell and bringing it into a larger cell to break down or digest or to defend against. And a big example we have for your microbial animal-like protists is paramecium. So that's what this picture is on the right. This is just a paramecium in a pond. Then we have your fungi-like protist. So these are your protists that share a lot of conditions and traits like your fungus. These are called heterotrophic, meaning that they have to eat other organisms in order to get their nutrients. Oftentimes they're going to resemble fungi, again, making them fungus-like protists. And oftentimes they'll form these sort of colonies of cells and produce spores to reproduce. So on this, on the right side, I have a picture of a slime mold. This is not a microbe per se, but I just wanted to give you an example of what a fungus-like protist looks like and how it's very similar to protistas. A micro example that I couldn't find a good picture of are called ambicomplexans. These are a parasitic fungi-like protists that actually can infect mosquitoes and release spores for reproduction. They actually are capable of both aiding and harming mosquitoes, which is really interesting. But again, shows the diversity of how protists can live in a lot of different environments. Then we have your plant-like protist. So as the name suggests, these are very similar to plants. These are always going to grow in water, always going to be in aquatic environments, and like plants, they're going to utilize photosynthesis to make their energy. An example of this we have on a microbe scale are called your diatoms, as well as we have organisms like your dinoflagellates. So very aquatic environments, very they are microscopic. Watch that word. And again, they can be food sources for other small organisms. As I alluded to earlier with the fungi-like protist, we do have some protists that are parasites, so they can make people very sick, besides just being other organisms that live in its environment. An example we have of this is with malaria. So a lot of people think that malaria is actually caused by mosquitoes, but that is actually not true. It's caused by a parasitic protist called Plasmodia. These Plasmodia is actually a protist that can infect your red blood cells and cause them to be deformed and cause a lot of other problems down the line. The reason people associate with mosquitoes, however, is that the mosquito actually carries the Plasmodia parasite. The mosquito is just a vector. It's not actually the one causing the malaria. It's just the one delivering the package, so to speak. But your Plasmodia is one of your primary examples of how protists can be parasitic and cause issues for the body. Lastly, we're going to talk about viruses. So as I mentioned at the top of the presentation, viruses are not exactly living organisms. In order for something to be considered living within the definition of biology, it has to do two things. It has to be able to, one, metabolize energy, so break down some kind of molecule into a smaller component to be able to get the energy to do stuff. And it has to be able to reproduce. It has to be able to make more of itself or copies or pass along its genetic information in some capacity. By themselves, viruses cannot do either of these things. However, if a virus is to infect a host and use their cells, they can accomplish both of these feats. So they can use a host cell's machinery to make more of themselves, and they can use the host cell's machinery to make energy for themselves. But by themselves, they're not exactly living, but if they're using an organism, they are considered living. So sort of a complicated outside the lines example. But viruses themselves, there's a couple key components to them. We have what is called a capsid, which is this green hexagon I'm drawing around. This is basically just a protein container that contains the inner workings of the virus, which is just genetic material. So it could be DNA, it could be RNA, it could be a little bit of both. And then sometimes you're going to have a plasma membrane that's engulfing the capsid. This doesn't always happen. It kind of varies virus to virus, but at the basic level, a virus is really just the protein container, the capsid, and the genetic material. So in order to reproduce, you're going to have the virus get to whatever host cell. Could be an animal cell, could be a fungus cell, depends on what the virus is. So virus will get to the host cell, that protein capsule will fuse with the membrane and deposit that DNA, RNA, in this case, we'll just say DNA, into the cell, and that will make its way to the nucleus, the control center, which has all of the DNA for the host. The viral DNA is going to do two things. First thing is that's going to replicate itself using the cell's machinery to make more of itself. So it's going to basically copy its genetic material to make a bunch of other genetic copies of itself. That's all these green squiggly lines that you see me running my mouse over. And then additionally, it's going to use its viral DNA to take over the cell's machinery and make a viral mRNA. So if you remember from intro bio, your mRNA is your messenger RNA, and that will go out to the cytoplasm to a ribosome and then synthesize proteins with tRNA. That's exactly what's going to happen with the viral mRNA. But in this case, when it goes to the cytoplasm, it's not going to make proteins for the cell. It's going to make viral proteins to assist the virus. And these viral proteins are then going to make the protein capsids to then re-encase all of the DNA that's been reproduced. So since the primary components are just the DNA and the capsule, that's all the virus needs to have to then make its way out of the cell and then infect new cells. So viruses, because they can infect pretty much anything and everything, can be responsible for a lot of different health problems. So a lot of diseases, both big and small, are contributed to viruses. So things like the common cold is due to viruses that we get throughout the year. Then we also have more serious ones like influenza as well as HIV. Some of these have given us a lot of issues. Some of these have been cured pretty easily. Really depends on the virus itself. And part of this complexity rises from whether the virus is a DNA virus or an RNA virus. So as I mentioned with the virus structure, a virus can have either DNA or RNA within its protein capsid, and DNA is much more stable than RNA. DNA has a basically a fail-state to correct for any mutations in the DNA. And these viruses tend to be much more stable. So since they're stable, they're not changing a lot, we can sort of get a good read on what the DNA sequence is and develop the appropriate vaccine to fight this infection for the DNA, since we know what we're dealing with. RNA viruses, however, are much more complicated. This is because they don't have a mechanism for checking the sequence. So a lot of times there can be mutations and changes in the actual RNA code. When this change happens, we have a mutation, which means that the virus we were once dealing with is slightly different than it used to be. The coronavirus is actually a great example of this. We've seen mutations of the coronavirus, making it a little more difficult to develop treatments for. So because we have this mutation, it's hard to develop a vaccine because the virus is changing so much. That's all we have for the microbiology units. So I will see you next time for the respiratory system.